Hi, everyone. Thank you so much to the organizers for inviting me to be a part of this exciting meeting. My name is Tippi MacKenzie, and I'm a pediatric and fetal surgeon at UCSF. And I'm going to tell you about a prenatal enzyme replacement therapy clinical trial. It's a phase 1 international clinical trial. These are my disclosures. I have research funding currently from Novartis and BioMarin. I've received research funding from Ultragenyx, which funded the enzyme replacement therapy work that I will tell you about. And I'm on the scientific advisory board of a gene editing company called Acrigen. So the fetus used to be this mysterious, elusive being that we met only after birth. But in the 1980s, with the advent of ultrasound, we started being able to peek in on the fetus before birth. And this actually ushered in a whole new field of medicine called fetal surgery. And it was invented right here at UCSF by a brilliant pediatric surgeon named Dr. Mike Harrison. And today there are fetal treatment centers all over the world doing a range of operations, usually for structural or anatomic problems. So this is kind of the range of things we're able to do for prenatal therapy right now. And like anything in medicine, we're really moving from the world of open fetal surgery, which is what we're doing for things like spina bifida, to less and less invasive things like laparoscopic or fetoscopic surgery, catheter-based interventions. And what I'm really excited about is medical treatments for genetic conditions using things like stem cell transplantation or enzyme replacement therapy or gene therapy. And these therapies can actually be delivered right into the umbilical vein of the fetus using just ultrasound guidance, using a very common procedure that we do for blood transfusions routinely in pregnancy for patients who have Rh incompatibility between the mother and the fetus. And there are lots of maternal fetal medicine or obstetric doctors who know how to do this umbilical vein procedure. So as I mentioned, we already do routine prenatal diagnosis for anatomic conditions using ultrasound. But if you think about it, this is just the tip of the iceberg. There are a lot more genetic conditions in the fetus that could be diagnosed. And right now, we only really recommend carrier screening for a handful of conditions. And if there's a family history or other reason to test, we can test the fetus using chorionic villus sampling or sampling of the placenta or sampling the amniotic fluid. And for certain conditions like Down syndrome, there are blood tests that can look for the chromosomal issues in the mother's blood. Now, you may ask yourself, why would you want to diagnose a disease before birth when it wouldn't necessarily change what you would think about the pregnancy? And there are two main reasons for this. One is that early prenatal diagnosis can actually help you coordinate the postnatal therapy for the child. You can coordinate delivery, for example, at a specialized center that has the various specialists that the child might need. You can actually meet with those doctors and specialists during pregnancy and learn more about the condition and have a better idea of what to expect. And you can even get started on insurance approvals for costly therapies. And in some conditions, actually prenatal therapy might improve the outcomes. The fetus is a really wonderful environment for prenatal therapy. One is you can potentially prevent organ damage for conditions that start before birth, like many MPS diseases. The fetus has a very unique and naive immune system, so you can potentially induce what we call tolerance to certain proteins so that the child after birth doesn't develop an allergic reaction to a new protein. And there's also no blood-brain barrier, so the medicines that we give can potentially better cross into the brain and treat the neurologic problems. And this is actually a really exciting time to be thinking about prenatal therapy for genetic diseases because of this convergence, a unique convergence of technologies and ideas. So we can now do rapid genetic diagnosis for many diseases. We can even sequence your entire genome in a matter of days sometimes. The fetal surgery field, as I said, has really expanded and disseminated all over the world. And there are new medications and even gene therapies being developed for a number of diseases. So the number of techniques that we can use to treat genetic diseases before birth include the ones I've listed here. Stem cell transplantation can be used, and we're actually running another phase 1 clinical trial for blood disorders. Enzyme replacement therapy, which is what I'm going to be talking to you about today. And then there's a lot of really exciting work in development for gene therapy or genome editing for genetic diseases. These are just in animal models, and nothing is really ready for prime time yet for clinical applications. So I'll focus on enzyme replacement therapy for today. So you guys are all experts in MPS disorders, and so I won't give a background on that, but just to talk a little bit about why we might want to treat an MPS disorder before birth. Number one is sometimes fetuses with MPS disorders, most notably MPS 7, don't actually survive to birth. They get sick as fetuses, and we can follow them actually getting sicker and sicker on ultrasound. And sadly, there's nothing we can do before this trial for that. Number two is a number of children can develop antibodies against the enzyme, the recombinant enzyme replacement therapy, which their body sees as foreign because their body doesn't usually make it. And so by giving the enzyme before birth, you could potentially induce tolerance, so prevent the development of those anti-drug antibodies. Number three, you can potentially deliver enzyme to the brain before the blood-brain barrier has formed. And number four, you can potentially prevent organ damage, most notably the heart and the liver, which are some of the organs that are already damaged at the time of birth for some diseases. So we wanted to test this idea in mice, and so in my lab, we did an in utero enzyme replacement therapy experiment that was led by two really talented students, Russell Witt and Ki Nguyen. And what we did was we took mice with MPS 7 and we did the fetal enzyme injection, which we can do pretty easily in the lab. And then after the mice were born, we continued to give them recombinant enzyme every few weeks after birth. And at the end of the experiment, we analyzed for a number of different things like survival and organ damage, bone length, did they develop antibodies, and what were the neurologic outcomes. So this experiment worked incredibly well. It was sort of beyond our wildest expectations. And I'll show you some of the results here. One is that the mice with MPS 7 also died before birth, and we had much improved survival. We had improved histology, which is the level of organ damage that you can see on slides. And what you're looking at here is that in the untreated animal, there are these little white areas, which are the vacuoles that build up. But with the enzyme replacement therapy given before and then also after birth, we didn't see that. And you can see it also graphed here, where there were a lot more of these vacuoles in the untreated animals compared to the treated animals. We measured their bones, and we saw that they had improved bone length. We saw that the enzyme penetrated into the brain, and especially into the cells in the brain called microglia, which are the really important storehouses of this enzyme. And we also induced tolerance to the enzyme. So based on these interesting results, we wanted to develop a safe protocol for in utero enzyme replacement therapy. And the heroes here were Dr. Paul Harmatz, who's an expert on MPS and lysosomal storage disorders. He's one of my colleagues at Children's Hospital, Benioff Children's Hospital, Oakland, as well as Marissa, who's a surgery resident, a student working with us. We had conversations with national and international experts. We surveyed patient groups such as you, the MPS Society, as well as other patient groups around the world. And we were able to get an investigational new drug approval from the FDA last year, and we're launching our clinical trial now. So the way that this trial will work is that we will anticipate having a diagnosis, a genetic diagnosis of the condition, either with the chorionic villus sampling or amniocentesis. And the FDA did give us permission to treat a number of different disorders under the same clinical trial. So for the MPSs, we'll be able to treat fetuses with MPS 1, 2, 4A, 6, or 7. Basically, all of these are the related diseases for which there's a postnatal FDA-approved enzyme replacement therapy that's given through the vein. We'll be giving the treatment through the umbilical vein every 2 to 4 weeks between 18 to 35 weeks of gestation. And then after birth, the patients will get the standard of care. So standard enzyme replacement therapy. And for conditions, if they were to get a stem cell transplant, they would also get that if the family chose to do that. And then we'll do a yearly follow-up for 5 years. So what are our endpoints? So it's important to keep in mind that this is a phase 1 clinical trial, which means it's really the earliest investigational study. So we're really trying to just see whether this therapy is safe and whether it's feasible to do. You know, are we going to even diagnose these patients before birth? But of course, in order to do any clinical trial in the pediatric or fetal population, you have to have some hope also that you're going to improve the outcome. So although it's not a guarantee, we do hope to improve the outcomes that are listed here. One is decrease the GAGs, or the glycosaminoglycans, which are the toxic metabolic byproducts that build up in tissues, and you can diagnose them in the urine. So we'll be looking for that. We'll be looking at whether we induce tolerance to the recombinant enzyme. And so are we preventing the formation of those anti-drug antibodies? And we'll also be looking at long-term outcomes, especially doing formal neurologic testing every year on the children to see if we've improved that. So for any trial, it's important to really understand who could participate in the trial. So these are the inclusion criteria. So it's fetuses between 18 to 35 weeks gestation. And this is really partly practical and partly, based on the scientific rationale. We're not really able to do a safe injection before 18 weeks gestation in the fetus. The umbilical vein is too challenging to find. And before 18 weeks, we don't anticipate most of the fetus would get diagnosed anyhow. And then after 35 weeks gestation, it doesn't make sense to do any other procedures. You might as well deliver the child and give the enzyme replacement after birth. And we definitely want to make sure that we have confirmed the presence of the lysosomal storage disease by genetic testing, either by CVS or amniocentesis, and also that we've confirmed that that particular genotype is going to give us severe enough disease so that it makes sense to treat before birth. And then there are also what we call exclusion criteria, where we don't think it would be safe to enroll a patient. And those are patients that have any other major other problem, like a structural problem that we can see on ultrasound, or a chromosome problem like Down syndrome, or any other major genetic mutation. And also maternal conditions that just wouldn't make it safe to do the fetal intervention. So fetal surgery is a really unique field where we have two patients, right? We have the mother and the fetus, and we want to make sure that maternal safety is really, paramount. And so we want to make sure that if there's anything that's going to make it too risky for the mother to undergo the therapy, that they would not be included in the trial. So the way that the trial will work is that if we have identify a fetus with an MPS disease anywhere in the country or even the world, we would do a video consultation with the family and the physicians. We've all gotten really good at doing that through COVID. And then if we think that they meet the inclusion criteria based on all the pregnancy and genetic information that we have, and if the parents are considering participating, then we would invite them to UCSF for the evaluation. We do have funding for all of the travel and the screening evaluation and the medical care involved with the trial. And then once we do the evaluation in person at UCSF, again, if we don't think that it's safe to enroll that patient, then they would be excluded, but then we could still help with organizing the postnatal care. But that if they meet the inclusion criteria and they would like to be included and they would like to participate in the trial, then we would do the in utero enzyme replacement therapy at UCSF. We do need to do the treatments at UCSF, and we would repeat every 2 to 4 weeks because of the half-life of the enzyme. And we've had patients participating in other trials who either stay in San Francisco for the duration of the pregnancy or who fly back home in between the therapies because, you know, you definitely have several weeks to go back and forth. And then delivery would be at home unless the family chose to stay in San Francisco, and that would be the routine postnatal care. But then once a year, we would invite the family back to UCSF to have the follow-ups, some neurological testing and other testing that would be really monitored by Dr. Paul Harmatz, who's our expert here. And then in conversations with the FDA, we've decided that it's really important to do a long-term follow-up because this really is the first time that in utero enzyme replacement therapy would be given for MPS disease. So we would be looking to see whether we've induced tolerance to the enzyme replacement therapy. We would be looking at the glycosaminoglycan levels. Those are the toxic metabolic byproducts. We would be looking at the mobility of the children as they grow, neurologic function, as well as growth. So for any therapy, of course, it's really important to look at the risk-benefit analysis. We've talked a lot about the potential benefits, but what are some of the risks of participating in this therapy? So one potential risk is an allergic reaction. We are hoping to induce tolerance, and that's what we've seen in any preclinical animal study, both with the enzyme replacement or introducing other proteins in utero. But there could be conversely an allergic reaction. Even a reaction in the mother, we think it's less likely because we are dosing directly into the fetus and it's a tiny dose compared to the mother's weight. But it's definitely something that we'll be monitoring very carefully for over time. Bleeding is a risk of any procedure that you do anytime. And then the other risk is preterm labor. We are injecting the medication directly into the umbilical vein, which, as I mentioned, it's a common procedure that we do for blood transfusions, but it's definitely, can be risky in unexperienced hands. This is how we do the procedure. It's ultrasound guided. And we have Dr. Juan Gonzalez on our team does all of our umbilical vein injections for transfusions and other medication administrations. We have reviewed our experience in more than 100 infusions, and we've had no fetal loss, and the risk of preterm delivery is less than 5%. But I think this is the most important risk to consider when you're considering participating in this trial. And it's important to really consider the feasibility of really doing accurate and timely prenatal diagnosis for patients with MPS diseases. We think that we're most likely to identify fetuses in families who already have a known carrier status, perhaps a known affected child, because we don't do universal screening during pregnancy for MPS or any lysosomal storage disease. And we don't do the screening because we don't yet have a therapy. But of course, if you have a viable therapy, that changes the equation for recommended screening. And with any new therapy, it's really important to engage the patient community. And we have surveyed the MPS Society. And so thank you for participating in the survey. And if you would like to fill out our survey, please let me know. We've really been overwhelmed by the positive reaction that we've seen for this in utero enzyme replacement therapy trial. And so just to give you some of the results, when we surveyed 169 parents, really mostly parents, 90% parents, and some patients with lysosomal storage diseases, on the likelihood of ending a future affected pregnancy, the majority have said of our patients or parents said that that was unlikely. Of choosing a fetal enzyme replacement therapy, if it were an established therapy, which it's not yet, of course, the majority of them said that they were likely. And then when we phrased it as, would you enroll in a phase 1 clinical trial for fetal enzyme replacement therapy? So this is investigational, no guarantee of success. Again, the majority said that they would be likely to enroll. So in summary, our clinical trial is now open for enrollment for people really anywhere in the world. And as I mentioned, we have funding to support the travel to UCSF, as well as the medical costs associated with the infusion. And if you would like more information, please let me know. You can email Billy, our genetic counselor, or myself. And I want to acknowledge the giant team of people who make this work possible, especially Dr. Harmatz, who's really our expert in lysosomal storage disorders, as well as Marissa Schwab, our surgery resident, who did a lot of incredible work on the FDA application and setting up the clinical trial. Billy, our genetic counselor. Juan Gonzalez is our maternal fetal medicine doctor, who's done a lot of work on the clinical protocol to make sure that we're really acknowledging safety for mom and fetus. Renata is another genetics expert. Teresa Sparks is another genetics expert, and Ramobia and Val are our program managers. So thank you, and I'll be very excited to take any questions and interact with you.